This is the activity you're going to do in cell and membrane transport. You're going to use your physioex CD, which is at the back of the book, or if you do not have the CD, you can actually go online to www.physioex.com. The first or second page of the inside cover of your book has an access code, so use that. So anyway, you've got to do about 4 activities from this physioex CD or the website. And this is the first activity, and I'm going to show you how you're supposed to begin. So what you're really trying to do is that these are 2 beakers. You're going to fill one with a solution which has solutes in it. The other one's going to be just water. So obviously there is going to be a gradient here because there's solutes here, no solutes here. And you're going to put a semi-permeable membrane, and which is this one. So I'm going to drag this over. You see that these semi-permeable membranes here have different sizes. This is 20, 50, 100 and 200. The one with the smallest size has smallest pores. The one with the largest size has larger pores. So obviously it'll be easier for substances to diffuse with the ones which have a larger pore size. But we'll begin with the smallest, and then in the left beaker we will put our solutions, either sodium chloride or urea or albumin or glucose. Use each of these membranes and then see whether solutes diffuse from the left to the right beaker. So you have to read the text before you begin every activity. So I'm going to do the first one for you, which is activity 1, and I'm going to use just one solution. You have to use all 4. So if you read your text, it says we have to increase this to 9 millimoles and dispense. So you can see it's dispensed in the left beaker. In the right beaker, we want to dispense deionized water. So now this contains deionized water. You can see none of the solutes, whereas this one has sodium chloride. And then we want to start. We want to keep the timer at 60 minutes. This is compressed time, and we hit start, and we want to see if the sodium chloride is able to diffuse through a membrane with 20, where the pore size is 20, and go to the right beaker. So I hit start, and we'll wait for the timer to come down to 0 when we'll know whether it has diffused or not. Now you cannot see in this, you cannot see any visible diffusion, but we still have to wait. So let us wait. It's down to 32, and we'll wait till it comes to 0, and you'll get a message, a small window, which will tell you whether equilibrium has been reached or whether diffusion did not occur. So you're going to do this for sodium chloride, and all of these you're going to use all the membranes and record your activities on the chart in your book, so that then when you answer questions, you know. So here it says sodium chloride, no diffusion. Hit OK. That means this membrane of 20 MWCO didn't allow sodium chloride to go to the other side. See, it remains at 9 here and remains at 0. You can record data, so you can see that there was no diffusion. Now I'm just going to put this back and just use the other one just to show you. So let's do 50, and we hit start again. Okay, you need to flush, and let's again go back to 9, dispense and flush, deionized water and dispense, and we hit start. Now your books tell you to, with the 20 NWCO, you flush each time, and you use urea and albumin and glucose. You can do it in any order. I'm just kind of showing you this, so that's why I had to flush in between. And you can see up here, look at this, sodium chloride has come to 4.5 and 4.5. So equilibrium has been reached. So that means sodium chloride diffused from left to the right beaker. And you can see this equilibrium at 10 minutes. The time is not important. What we see is that 50 MWCO allowed sodium chloride to diffuse from the left to the right beaker, and we can record data again. And you can see um, with 50 you can see average diffusion rate was 0.0150. We are not interested in the diffusion rate in this particular activity. What we want to see is, does the MWCO allow diffusion to occur or not? So now you're going to use all these, so you can put this membrane back, use flush, and then use all the membranes. So similarly, you'll be doing this for urea, albumin, and glucose, and see which of these membranes allows it to pass from one side to the other. Now, after having done this experiment, the next one you're going to do is facilitated diffusion, where you're using sodium chloride and glucose. And in this, you use transport carriers. You use those. So read your text. Again, the principle is the same. You want to see whether it diffuses from the left into the right beaker. And, you're using these membranes and you're using these glucose carrier proteins. The next one you're required to do is osmosis, is the activity on osmosis, where again you look at sodium chloride, albumin, and glucose. And I will show you one where now I'm bringing the 20 MWCO. It tells you to increase the concentration to sodium chloride concentration to 8 on the left side, and you do deionized water and dispense deionized water on the right side. You set your timer at 60 and you hit start, and then you are expected to watch the pressure change. So if substances diffuse from the from the left to the right, obviously no pressure will be generated, because you can see that equilibrium is reached. But here, if you notice that the pressure is increasing and stopped at 272, and if you remember from the first activity, 20 MWCO did not allow sodium chloride to go from the left to the right. So the solutes remained over here and they didn't go across, and we'll wait till this comes to 0. And hence you can see that pressure is generated, because this is a closed space, and it's not allowing substances to diffuse from the left to the right. So actually it's generating pressure, which will try to bring water in. So you can see osmotic, no diffusion, osmotic equilibrium reached over here. So now let's flush. We'll flush this, and I want to show you. So 20 generated pressure for sodium chloride. Let's bring this 50, and we again take this to 8, dispense, deionized water, I dispense, and the time is the same. I hit start, and notice, you'll see that no pressure is being generated here, because if you remember, 50 allowed sodium chloride to go from the left to the right. So if substances are able to diffuse, then pressure is not generated. So that is what you're trying to do in this activity. And the last activity you're required to do is active transport, which is this one. This is pretty simple. In active transport, ATP is one of the, remember ATP is used, so you dispense ATP, and then you also use glucose carriers and sodium and potassium pumps, because that's required for sodium transport. So this is how you're going to use it. So you see whether transport occurs with the use of ATP, and when you don't dispense ATP here, see whether the rate is faster or slower. So this is how you do these activities.